of plasma membrane. To maintain a steady state and homeostatic intracellular environment, a living cell must be an open system. So it takes useful materials like water, oxygen, energy and nutrients which are essential for its survival. At the same time, it also expels carbon dioxide, nitrogenous wastes, feces and excess of energy as heat out of body to maintain a state of maximum efficiency inside the cell. Transport across plasma membrane. The internal composition of the cell is maintained because plasma membrane is a selectively permeable structure. The plasma membrane forms a barrier that blocks the free exchange of molecules between cytoplasm and the external environment of the cell. Most biological molecules are unable to diffuse the protein-free phospholipd bilayer. Small nonpolar molecules, such as oxygen and carbon dioxide, readily dissolve in synthetic lipid bilayer and, therefore, diffuse rapidly across them. Small uncharged polar molecules, such as water and urea, also diffuse across a synthetic lipid bilayer. By contrast, synthetic bilayer are highly impermeable for charged molecules. However, the plasma membrane, unlike synthetic lipid bilayer, allows the passage of various polar molecules such as iron, sugar, amino acids and others. Special classes of transport protein are responsible for transferring such solutes across cell membrane. Thus transport across plasma membrane can occur either through the lipid bilayer or through the transport proteins. The characteristics of lipid phase diffusion and protein-mediated transport are basically different. Transport across the plasma membrane is of two types. 1. Passive transport. 2. Active transport. 1. Passive transport. Passive transport occurs along the concentration gradient and without the use of metabolic energy. If a transported substance carries a net charge, its movement is influenced by both its concentration and the membrane potential, the electric potential across the membrane. The combination of these two forces, Cale's electrochemical gradient, determines the direction of transport of a charged molecule across the membrane. Thus an ion diffuses not simply down its concentration gradient but, more exactly, down its electrochemical gradient. Passive transport may be 1. Simple diffusion. During simple diffusion, a molecule simply dissolves in the phospholipid bilayer, diffuse across it. No membrane protein are involved and the direction of transport is determined simply by the relative concentration of the molecules inside and outside the cell. The relative diffusion rate of any substance across a pure phospholipid bilayer is proportional to its concentration gradient the layer and to its hydrophobicity and size. Movement of solutes by diffusion is always from a higher to a lower concentration, and the rate is described by Fick's law of diffusion. Where J is the flux per unit area, D is the diffusion coefficient and delta C is the difference in concentration between two regions separated by a distance delta x. The negative sign accounts for the fact that diffusion is toward the lower concentration. 2. Facilitated diffusion. Like simple diffusion, facilitated diffusion involves the movement of solutes along the concentration gradient. However, the passage is mediated by transport protein, carriers and channels, and is selective in nature. Facilitated diffusion may be carrier proteins and channel proteins mediated. Classes of proteins that mediate facilitated diffusion. Carrier protein mediated. Carrier proteins called transporters or permeases non-covalently bind specific molecule to be transported on one side of membrane. They then undergo conformational changes that allow the molecule to pass through the membrane and be released on the other side. A classic simple example is the movement of glucose mediated by carrier protein, glucose transporter, GLUT. There are various isoforms of the glucose transporter family. These carriers are composed of 500 amino acids and possess 12 transmembrane segments. Channel proteins mediated. Channel proteins form open pores through the membrane, allowing the free diffusion of any molecule of the appropriate size and charge. Rate of diffusion mediated by channel protein is higher than those mediated by carriers. Channel proteins concerned specifically with inorganic ions transport are called ion channels. Ion channels are highly selective. Most of ion channel are not permanently open. Some channels called voltage-gated channel open in response to change in electric potential, others called ligand-gated channels open in response to the binding of ligands. Many animal and plant cells contain specialized water channel in their plasma membrane to facilitate the water flow called aquaporins. These are a family of transport protein that allow water and a few other small uncharged molecules, such as glycerol, to cross membrane. The properties of ion channels can be studied by means of the patch clamp technique. This technique has provided lots of valuable informations about ion channels, and its inventors, Erwin Neher and Bert Sackmann, were awarded the Nobel Prize in 1991.